everyone, welcome to the next episode of my science podcast. Today it is an absolute honour to welcome Sir Paul Nurse, who was knighted in 1999 and was awarded the 2001 Nobel Prize in Physiology or Medicine for his discovery of protein molecules that control the division of cells. In 2010, he became the first director and chief executive of the Francis Crick Institute in London, and for 5 years was president of the Royal Society. In 2013, he became the winner of the Albert Einstein World Award of Science, conferred by the World Cultural Council, and since 2017 has been the chief scientific advisor of the European Commission, so it is extremely kind of him to give up his time to talk to us today. There we are, I think we're recording now. So thank you so much for joining us today. I've done my introduction already on another video, so we'll crack on straight on with the questions for time purposes. So my first question is, you have identified the CDC2 gene in the yeast Schizosaccharomyces pomp that controlled the progression of the cell cycle from G1 phase to S phase and the transition from G2 to mitosis. So may you expand upon how exactly this gene controls these transitions?
Yes, I can, and it's a pleasure to talk to you, Martha, about them. This is the CDC2 gene encodes a protein called a protein kinase, and a protein kinase is an enzyme which puts phosphates onto other proteins. And the consequence of that is that the phosphate changes the structure and properties of those other proteins. And those changes can alter the activity of these other proteins. You can either turn them on or turn them off. And so protein kinases act as switches that switch things on and switch things off. And this particular one, the CDC2 protein kinase, which my lab discovered in fission yeast, you rightly described, but actually is found in every eukaryotic cell from yeast to humans. So what I'm going to tell you is what happens in every eukaryotic cell. What it does is it phosphorylates different substrates, different proteins at different stages in the cell cycle, starting in G1 at the beginning of the cycle, which leads then to the replication of DNA, the copying of DNA during S phase, and then other events in the cell cycle after that, which lead to the initiation of mitosis from the G2 period into what's called M phase. So this protein kinase, this CDC2 protein kinase, which incidentally is called a cyclin-dependent kinase, is a molecule that controls all the events of the cell cycle from the very beginning to the end. And it's because of this rising activity through the cell cycle that we get all the ordered events of the cell cycle that eventually lead to a proper cell division.
Oh, wow. Okay. And so how did this, your research, then lead to improvements in the knowledge of retinoblastoma and breast cancer?
Well, that was quite a long story. As I just explained, this particular protein and the other proteins that regulate it are found in all eukaryotic cells, including humans. And it turns out that when you when cells in your body undergo cell division, they all require this key regulator. And in cancer, which is uncontrolled cell division, this process is, this protein kinase is activated so the cells can undergo cell division. And it turns out that in breast cancer, if you add inhibitors of this particular type of protein kinase, then that can have a differential effect on breast cancer cells dividing. And when used in combination with other drugs, can actually control cancers, breast cancer, and maybe other cancers as well, in fact, so that you can use it as a therapy. It's not what I started out the work to do. I wanted to understand the fundamental process, but I'm obviously very pleased that it's turned out to be quite useful for cancer patients in particular breast cancer patients.
Absolutely. So my next question is, probabilistic cell size homeostasis is driven by noisy expression of cyclin B in fish and yeast. And so how is this linked to the MEGA2 gene, which senses size and corrects for any deviations by advancing or delaying onset of mitosis?
Well, the short answer to that question is I don't know. But I can explain a little bit more about what's going on there. And the reason I don't know is that the MGA2 gene you just mentioned has only just been discovered, and I've only recently published it. It's never been described as to what it does in controlling the cell cycle elsewhere. So all we can do is speculate, which I will come to in a moment. So the first question was about noisy accumulation of cyclin. And what I didn't explain was that the CDC2 protein kinase is actually part of a complex made up of two proteins, the protein kinase and another component called cyclin. And cyclin is the reason why it's called cyclin dependent kinase, or CDK. So there's two components, the kinase that does the phosphorylation and the cyclin which activates the kinase so it can work. That means that for an active CDK, you need to have enough of the cyclin present. And that accumulates gradually as cells proceed through the cell cycle. And what we discovered was that the accumulation of that protein was a bit irregular. It was noisy. So, and that noisiness meant that the amount of CDK activity in the cell is also noisy. It's also variable. And that in turn means that when cells go into mitosis, the timing of that is also noisy, that is variable. And so the whole thing is not as precise as you could imagine, and we suggested is due to the noisy accumulation of the cyclin activator. Now you mentioned MGA2, and MGA2 is a gene, and we don't know very much about it, but it increases the noisiness of going into mitosis. So probably, as you assumed in your question, these are related in some way, but we don't know what the relationship is. I can offer a guess, and the guess is because MGA2 influences the manufacture of lipids in membranes in cells, and because we know CDKs enter the nucleus through the nuclear membrane, which is made of lipid, we have speculated that maybe in an MGA2 mutant, the transport of the CDK containing the CDC2 protein kinase from the cytoplasm into the nucleus is very varied and noisy in an MGA2 mutant. And that in turn means that the activation of the kinase is noisy in the nucleus as well. And maybe that's the explanation. But like in all science, we have these ideas, and sometimes they're right, more often they're wrong. And so this is probably wrong, but over the coming months and year or so, we hope we can get better insight to see if it's correct or not.
Absolutely. And that brings us on nicely to my next question, that the endoplasmic reticulum protein LNP1 compensates for a lack of LEM2, acting as a secondary barrier to membrane flow, maintaining the nucleocytoplasmic ratio. And so other than this nucleocytoplasmic transport, how can lipid biogenesis and RNA processing play a role in this?
Yes, so this is also complicated, and I also am not sure of the answer here, but I can tell you a little bit about what is happening here. What we were interested in was what determines the size of the nucleus. It's a problem which is very easily stated, but actually one that has hardly been studied even in the last 100 years. So I decided we should have a look at this as a problem. And what we discovered was that the nucleus is exactly proportional to the size of the cell in which it's in. In the case of fission yeast, and you've already gathered, I work nearly all the time only on fission yeast because it's easy to work with and has fantastic genetics. In the case of a fission yeast cell, the nucleus occupies 8% of the space of the nucleus. And that is kept very constant. So we looked for mutants in genes that would alter that ratio, which would either make the ratio bigger or make it smaller. And you mentioned a couple of genes like LEM2. And if you delete the LEM2 gene, then what happens is that the nucleus gets bigger. It goes from 8% to about 12%, or maybe even a bit higher. So these proteins have some influence on the size of the nucleus and how that's regulated, which, as I already said, is a completely unworked on problem. So this is just the very first observations, or nearly the very first, that could that could explain it. What we speculated, because we know that LEM2 influences the transport of membrane from one organelle to another, and in particular into the endoplasmic reticulum, which is other membrane structures in the rest of the cell. That what we speculated was that the size of the nucleus is kept at 8% by regulating the transfer of membrane in and out of the nucleus from other components, membrane components within the cell, like the endoplasmic reticulum. So we speculated it's like a sort of valve, that if the nucleus is too small, you open the valve and you get more membrane flowing in. And if the nucleus is too big, you close the valve and you switch off membrane from going in. And as the cell is growing, of course, that will keep nuclear size homeostasis. This is also a speculation, and this is also not clear whether it's correct or not, but that is, I shall repeat, how science works. We do experiments, we make observations, we come up with a hypothesis, and then we test it to see if it's right or not. And I repeat, most often it isn't right, but you use that to make progress as to work out what might be right.
Exactly, and that's what makes science so exciting.
It does indeed.
So my final question is, how did you order cyclin-dependent kinase substrate phosphorylation, and what did these experiments suggest?
So I've explained already that the hypothesis, which I think is correct, so this is much more sounder than what I've just been speaking about, is that the cyclin-dependent kinase increases in activity as cells proceed through the cell cycle. And because the CDK can phosphorylate many substrates, actually hundreds, probably 400 or 500.
Oh, wow.
So it's very many substrates, which is unusual for a protein kinase. It phosphorylates only some of those substrates early on in the cell cycle, substrates that have to be phosphorylated if you're going to go from G1 into S phase. And then it phosphorylates other substrates, actually rather more, in G2 when the cell goes into mitosis. So what we did to test whether that was correct was to make cells go through the cell cycle in a synchronous fashion, so that you had all the cells going simultaneously through G1 into S and then later G2 into M. We extracted the proteins, and then we analyzed the proteins on a machine called a mass spectrometer. And that allowed us to detect which of those proteins were phosphorylated, because the machine can do that. And what we found was that substrates which are required at S phase can get phosphorylated right at the beginning of the cycle when there's very little activity with the cyclin-dependent kinase. But substrates required at the end of the cell cycle couldn't be phosphorylated then, because they needed much higher activity to become phosphorylated. So this is really a very simple model in actual fact. All it is CDK activity, cyclin-dependent kinase activity rises during the cycle. It phosphorylates some proteins early on, which can be phosphorylated easily, and that brings about S phase, and other proteins which are phosphorylated with greater difficulty and so need higher activity, and that only occurs at the end of the cell cycle when the cell undergoes mitosis.
That's wonderful. Thank you so much for this. I really appreciate your time, and I'm extremely grateful for this. So
not at all. I hope it all made sense.
Oh, it definitely did. Thank you so much.
Not at all. Okay, So good luck with your podcast, and good luck with your studies. Do you want to be a biologist?
I do, yes. That's my hope for the future.
Well, good luck with that too. All right. So
Thank you
bye-bye for
very
now.
much. Thank you.
Bye.